Hi, everybody. My name is Will Samuel, and I'll be presenting my senior thesis project on the Cripple Creek restoration in Fairbanks, Alaska. Chinook salmon abundance has been decreasing across the state of Alaska over recent decades, and especially in the Yukon River drainage. For those of you who don't know, the Yukon River stretches from western Alaska across the state into the Yukon Territory of Canada. And Chinook salmon within this river affect a variety of subsistence and sport fishing stakeholders. And these fish also contribute important marine-derived nutrients to freshwater ecosystems. Rivers in interior Alaska have been following that same general decline over recent years. Specifically, the Tanana River drainage in interior Alaska, which contributes about 15% of Chinook salmon to the Yukon River stock, has seen a decline since the 1990s. You can see here that the Chena and tshawytscha rivers, which are within the Tanana River drainage, have seen low escapement in recent years. One response from managers has been a habitat management initiative located in the city of Fairbanks, Alaska. This is meant to improve and expand rearing habitat for juvenile life stage Chinook salmon. So today we'll be looking at a case study restoration located in the city of Fairbanks, which is nested in the Chena River drainage. You can see Fairbanks there is on the western portion. And specifically, we'll be looking at the Cripple Creek watershed, which is on the very westernmost portion as a rather small catchment. And we'll be looking at just one section today where a recent restoration occurred. So a little bit of background about this area. Back in the 1930s, a placer mining operation diverted water out of the original creek channel, which you can see here in light blue, and then dug a drainage ditch alongside of that, which you can see in dark blue, and then diverted water out of the original creek channel into that drainage ditch in order to move sediment away from their mine site to access the gold-bearing ground. This drains directly into the Chena River main stem and is pretty poor quality fish habitat. It's channelized and silty. It's lacking in riparian cover in some areas. So in 2015, U.S. Fish and Wildlife underwent a restoration project to repair this area. That included a series of culvert replacements and retrofitting near the barriers to fish passage. And once these projects were complete, in fall of 2020, they used the Happy Creek channel, which you can see here in pink, to redirect water out of the drainage ditch back into the original channel. So this spring, once everything thaws out, water will once again flow through the historical Cripple Creek channel after almost 100 years. This restoration added about 1 kilometer of linear channel habitat to the creek, but more importantly, it's better quality habitat for fish. So throughout this whole process, the Fairbanks Soil and Water Conservation District has been undergoing a data collection effort in order to establish some baseline environmental data during the restoration. So from the summers of 2018 through 2020, the Fairbanks Youth for Habitat Corps, which is their kids' summer camp, periodically operated minnow traps on both the original creek channel and the drainage ditch. And this is what made up the bulk of the data for my project. There's a pretty diverse and widely distributed fish community in Cripple Creek, which is encouraging to see so close to Fairbanks suburbia, and especially in this impaired waterway. And although this restoration is intended to expand rearing habitat for Chinook salmon, the Chinook salmon aren't present in any great number in Cripple Creek at this time. So we decided to use the two most abundant and widespread species in Cripple Creek, which were Lake Chub and Longnose Sucker. So with that, we wanted to use fish metrics like abundance, length, and distribution to see how successful this restoration was. And while we were at it, we wanted to incorporate some environmental variables like river discharge and water temperature. We then divided the creek into sections using the culvert replacement projects as boundaries for those sections so that we could assess fish passage. And then we calculated a standardized weekly catch per unit effort and mean length so that we could compare data between years. We then used those data as inputs for a series of analysis of variance or ANOVA tests and linear regressions. And then we used those tools to conduct a spatial and temporal analysis on fish abundance and length in Cripple Creek. And then, of course, incorporated our environmental variables as well. Some of our results, we found that Lake Chub catch per unit effort decreased significantly from 2018 through 2020, which was kind of surprising and disappointing. Typically, after a restoration, you would hope that fish abundance increased significantly afterwards, but we saw the opposite here. And we believe this could be because I spent a lot of extra time out there trapping this summer. I tried to fill in the the holes and data gaps through sampling so that we could have one continuous data set from May all the way to the end of August. But that may have backfired a little bit, and we may have actually oversaturated our sampling sites, and that may artificially deflate our catch per unit effort as a representation of fish abundance. And then another possible explanation is that we had really record high discharge in 2020 in both the Chena River and Cripple Creek. So there's a potential that high flows might decrease the efficiency of our traps, and that could be playing a role as well. And we found that Lake Chub in the upper sections were significantly longer than those in the lower sections. And this supported one of our hypotheses. We thought that if those culverts were actually acting as barriers to fish passage, that they would discriminate against the small fish first, that those poor swimming fish would have a hard time traveling through those barriers before the large fish did. And that's what this data shows. And we found that Lake Chub catch per unit effort was negatively associated with discharge. And this was surprising. We expected under high discharge in the Chena River main stem, which is what you see here, we expected fish abundance in Cripple Creek to go up. We thought that they would move out of the main stem river and utilize that tributary habitat as refugia during those times, but that's not what we see here. So we think that fish might either become less mobile under those high flow conditions, or it simply just decreases the efficiency of our traps. And we found that longnose sucker mean length was negatively associated with water temperature. And this was also contrary to one of our hypotheses. We expected fish size to be correlated positively with water temperature because of either the higher metabolisms under warm water causing growth, or the fact that fish might be using that tributary habitat as thermal refugia during hot conditions. But that's not what we see here. And again, it's not correlated super strongly. So this may have been statistically significant, but it may not necessarily be biologically significant. And we found that weekly mean length of these fish increased throughout the summer of 2020. So once you account for outliers for longnose sucker, both fish have a positive trend line for mean length. And we can't necessarily attribute this to individual fish growth because we didn't actually tag and track any fish throughout the summer, but it does kind of support the idea and the intention of the restoration that this tributary habitat can be used as rearing grounds for these juvenile and small fish throughout the summertime. And then finally, I'd like to mention the Chinook salmon that I caught all the way up at Site 16. This is only the third Chinook salmon ever caught in Cripple Creek, which was pretty exciting. And both the other fish were caught right near the mouth of the Chena. So it was pretty cool to see that this guy had navigated up through three pretty significant barriers to fish passage and made it all the way to our very top site. And it was a large and healthy fish. So although it was just one, if one can do it, hopefully more can. And in the future, this creek begins to be populated with more Chinook salmon. So our results are somewhat inconclusive, but analysis is ongoing. We're still kind of trying to pick everything apart. Interestingly, Lake Chub gave us most of our significant results, and we think that's because there are potentially more mobile fish species, and they may be a better metric for estimating these environmental or physical effects on salmonids because they're more physiologically similar. And then I'd like to note that it's kind of difficult to establish causal relationships with these trends just because of the minnow trap data collection method. And then with that, we might recommend in the future that the experimental design and methods collection planning be reassessed, because most of this sampling was done with convenience sampling, both temporally and spatially. So it might be better to reassess your data collection methods if you're looking to estimate some of these complex ecological relationships. So there is future research planned in Cripple Creek. U.S. Fish and Wildlife has a fish occupancy study planned after they collect another 3 to 5 years of post-restoration minnow trap data. So hopefully that will kind of build upon my project and bring in a little bit longer term data set to look at the effects of the restoration on Cripple Creek and those fishes within that waterway. And beyond that, this Chena Watershed Resources Action Plan is that habitat management initiative that I spoke about at the beginning of the presentation. This has basically been a collaborative effort between about 15 different agencies and organizations in Fairbanks who have all come together to try to find a unified way to manage the aquatic resources and watershed attributes within the Chena River drainage. And so you can see this map to the right is one of the products from that working group. And basically what they did was prioritize which areas were of high conservation value, which areas get you the most bang for your buck, and which areas are the most impaired or with the most limiting factors. So you can see those dark green and dark blue areas are the highest priority for conservation need, and a lot of those are made up of restorations very similar to the Cripple Creek Project. So hopefully my project can help to inform some of those future planned restorations and just overall habitat management within the Chena River drainage. And all of this habitat management definitely plays an important role in salmon conservation within the Chena River drainage. Like I mentioned, Chinook escapement within the Chena River has been decreasing pretty drastically over recent years. So it's important for us to provide access to those high quality rearing grounds and minimize our human impacts on this fish species. And by doing that, we can increase recruitment into the marine fisheries and help contribute to the overall viability of the Yukon Chinook salmon stock. With that, I'd like to thank everyone who contributed to this project. And I'd be happy to take any questions and feedback.